All right, I'm going to go ahead and get started while the last few people grab their lunch. So thank you all for coming on this Good Friday, and it's spring break, so our parking decks were nice and empty this morning, and then I thought, hopefully people will show up, and you are here, so I am very glad to see you. So today I'm going to talk about target validation. It's an aspect of drug discovery that's many times, well, experienced people notice very important, but as an academic investigator, many times we just assume that what we're working on and our target is the best thing since sliced bread. So today we're going to talk a little bit about what goes into validating a target. And just full disclosure, so I'm the presenter today, but Dr. Mark Sudo, he's the Vice President for Drug Discovery at Seldon Research. He's presented this in the past, so I actually pilfered a lot of his slides. So if you have any questions, I don't know the answer, I can ask Mark, just so you know. All right, so this is just to refresh your understanding the drug discovery and development pipeline as it's many times depicted. But on the left side of the slide, it starts with target identification. So we'll talk a little bit about that today. Hit generation, lead generation, so finding chemical matter that seems to have drug-like properties that may ultimately become a drug that you would then test in preclinical models in animals before you go into clinical trials that with their phase 1, 2, 3, hopefully then end up with FDA approval, and after that you can actually sell the drug to your patients. But how do you identify a target that's of interest? And I've touched upon this a little bit in the overview, but just to make sure we're all working on the same definition, the target is actually the naturally existing cellular or molecular structure involved in a disease that the drug in development is meant to act on. So it's really, what does this drug bind to? What does it physically interact with to do its job? In order to understand that, to identify these targets, you really need to understand the molecular mechanism of the disease in as much detail as possible. So, for example, you could identify that a certain signal transduction pathway is absolutely critical for a certain disease, but that's not specific enough because a signal transduction pathway involves many enzymes, receptors, maybe signaling molecules. So what in that pathway do you actually then want your drug to bind to? That's almost a deeper level of understanding of this biology. So you really need to do a lot of basic research and understand the basic biology before you can identify a target that's hopefully going to result in the development of a new drug. And I've already also shown this picture before this is another iteration of the drug discovery and development pathway, but on the left, this is assuming you do high throughput screening. Everything in blue is really easy. So compound libraries, you can just buy them. Target identification is actually fairly easy as well to identify, to come up with a new idea and a hypothesis. And then if you use robots, the high throughput screening itself and the robots execute this is fairly easy as well. But in everything in gray is a bottleneck in drug discovery. So here actually target validation is indicated also as a bottleneck. And so I'm going to talk today a little bit about why it is a bottleneck, because it is pretty hard to predict from idea of identify this new target to figure out is this actually going to make a difference in patients, yes or no. And that kind of ties into with the success rate in the clinic slide I've shown before as well. When you look at how many programs go from a preclinical phase to phase 1, it's actually about 64%. So it's, not 100% of course, but it's pretty good. 44% of molecules go from phase 1 to phase 2. So phase 1 is typically really looking at toxicity and tolerability of these molecules in humans for the first time. Where the real drop-off is from phase 2 to phase 3. Only 22% of molecules get out of phase 2 successfully move to phase 3. Now, what you study in phase 2 is, does my drug work in a patient population, yes or no? So this is typically the first time you really ask this question of, is my target a good one? And you can kind of see that this is a really hard question to answer earlier. We can hypothesize till the cows come home, but it's not until you do the actual human experiment that you really know if your target is validated or not. So just to show you some typical numbers from large pharma. So let's say they start about 100 new screening programs or assays around new targets. Typically only about half of those actually find acceptable lead molecules or chemotypes, so scaffolds on which you can build many different molecules to pursue. So half of them don't actually even get there. But those 50, then you find a scaffold, you find a lead molecule, but then only about, again, half of those, so 20 to 25, actually end up in late stage lead optimization. Of those, only 10 advance into phase 1. So from those 100 that you started with, even only a tenth made it actually into the clinic. And then, going back to this slide, showing what the success rates are in the clinic, you can see it's actually a pretty long shot to find a target that actually works in patients. So why is that? There's not a golden answer to this, but I'll touch upon a few aspects, hopefully today, that may help us make some better decisions, even though ultimately, like I said earlier, the final experiment is the human experiment, is clinical trials. You really don't know before then. So why do compounds fail? So it used to be that pharmacokinetic parameters really were one of the main reasons why drugs, when they finally went into humans, didn't make it. So until, I'd say, maybe 10, 20 years ago, there was really not a good way to evaluate what the human pharmacokinetic parameters would look like. So the acronym ADME stands for absorption, distribution, metabolism, and excretion. So all we had at the time was do some rodent studies, see what's the pharmacokinetic in mice, rats, maybe do some dog studies, maybe do some monkey studies, and then ultimately go to humans. The translation is not always a direct one. So now with human liver microsomes and more advanced modeling and so on, we do have a little bit better sense of what it takes to have good pharmacokinetic parameters in a human being. Again, you don't know for sure until you test it, but our predictive power has really increased over the last couple of decades. So it sometimes causes a compound to fail, but it's not the main driver anymore of failure. Now, of course, sometimes you come against toxicity that's in humans that was just not predicted by animal studies. We are a different animal, so sometimes you just don't know. Again, we're getting much better to predict that compared to, let's say, 50 years ago, but sometimes you don't know. Now, there can also be commercial reasons why companies decide to abandon a program. For example, Pfizer closed down its neuroscience unit about 3 months ago. So they had huge programs, and they just said, we don't want to be in neuro anymore. So they closed down their unit. So I'm sure they had good reasons for that, or I hope so. But these are for-profit companies that invest a lot of resources in these studies. So they are sometimes driven by commercial decision and not necessarily by whether it's a good idea or not. But most often now, as was also illustrated by this attrition rate in phase 2 trials, is that the drug just doesn't work. Lack of efficacy in humans, lack of efficacy in patients. So it could be that the biological rationale is just incorrect. We still don't know a lot of biology. We think we have a pretty good pattern and understand with all the technologies these days with proteomics and all that kind of stuff, but it's still a big, large black box for the most part, and especially when it comes to give something to a human and see what happens. There's just a lot we don't know. So it's not like an engineering problem. And I think sometimes you hear some tech companies, and Google actually did the same thing. They said, let's just use our prowess in engineering and apply it to drug discovery, and we're going to use technology to drive the discovery of new medicines. It's just those chemists and biologists are just a bunch of amateurs. They don't know what they're doing, and if we just use our engineering prowess, we're going to fix this problem. Well, they tried to do that, and what do you know? They didn't succeed. Again, because when you design a computer or a car or a space shuttle, for that matter, you as a human put together all these different pieces so you can use engineering to overcome solutions and so on. Biology, we didn't design it, right? Evolution or whatever your spiritual belief is, there's a lot we don't know as humans. We can observe, we can hypothesize, but we didn't put us together. So engineering is not really going to solve that problem. Now, of course, you can use engineering to optimize processes and use artificial intelligence maybe. So I'm not saying there's no place for it, but the biological rationale, you can use engineering to maybe come up with hypotheses, but ultimately it's not until you do the experiment that you know. Another big problem is the relevance of animal models, and I touched upon it a little bit in the overview talk as well. But here's just 2 examples in oncology and Alzheimer's. We have had great success in finding cancer drugs and Alzheimer's drugs that work in rodents. But when you do the trial in humans, they just don't work. And that may be because the animal model doesn't reflect the human biology. And many times also we design animal models based on the hypothesis that we have how the pathway works. So you're kind of engineering it to give you the answer you want, right? So let's say you make a particular type of knockout mouse because it shows a certain phenotype. And then you treat it with a drug that interferes with the pathway. Well, you may see a good outcome in that particular knockout mouse, but in humans, it's a different situation. So this is just a headline of a paper that was in a European Drug Target Review article in 2014, where they talked about validating molecular targets in preclinical drug discovery. And they analyzed a lot of phase 2 and phase 3 trials in 2012, 2013. And about half of the drugs failed due to insufficient activity. So in other words, the clinical target validation fails about 50% of any therapeutic approach. So again, an in-depth biological understanding of the target is helpful, but ultimately you don't know until you try. There's another really good article that looked at AstraZeneca's drug pipeline. It was actually by AstraZeneca itself. It's in Nature Reviews Drug Discovery 2013. So it's called Lessons Learned from the Fate of AZ's Drug Pipeline, a 5-dimensional framework. And what they evaluated was what's the success rate of driving a program through all the different phases in their pipeline. So from early stage research projects to preclinical development to phase 1, 2, 3, etc. And it turned out that many times, as in these companies that probably, not just AstraZeneca, but a lot of companies, bonuses and so on were driven by how many projects did you successfully get over the finish line of your particular unit. So let's say your preclinical development unit, you would get rewarded for getting things through your particular piece of the company and hand it off to the clinical development people. Well, so you're rewarded for getting things over the finish line, but that finish line is just the short-term finish line, right? Get it into the clinic. So the people in that particular unit are driven to make that a success, hand it off to the clinic, and it's somebody else's problem. So ultimately, that then resulted in a lot of projects being driven forward by these discrete different units, but kind of lacking the overall coherence of actually thinking strategically, is this even a good program to engage in, yes or no? It's a wonderful review. And what they finally said that they learned from this as AZ's management is the 5 R's, they called it. So before they start on the program, they want to identify, is it the right target? Is it the right patient population for this target? The right tissue? So for example, you can imagine it's a very different program if you need to go in the CNS versus in the lungs or in the liver, things like that. Right safety profile, and does it have the right commercial potential? Of course, this is a commercial company, so they need to look at this. And it's too short ago, 2013, to really evaluate whether the success rate has changed or not. But I think it's something that the whole industry is looking at to see them changing these paradigms in their reward structure. Will it change the outcome? And I think what they really want to encourage is people asking the tough questions and getting the right answer. And the right answer can be yes or no, this target is a good one, yes or no. The right answer should not necessarily be, can I get this drug into somebody else's hands within the company? That may not be the right question to ask. So what we run into a lot here within the Alabama Drug Discovery Alliance is the rationale that academics that have an idea that come to me and they say, I have this great target. It's in the literature, for 15 years, I was the first one to publish it maybe. We have all these publications, and if I inhibit this enzyme, for example, I'm going to cure cancer. It's going to be wonderful. And nobody has found a drug yet. And then, usually they first talk to me, and then I pull in Mark Sudo, the Vice President of Drug Discovery for Southern Research. And Mark then always starts to chuckle a little bit. He's like, well, if it's been in the literature for 15 years and there's no drug yet, probably companies have tried. There's probably a reason why there's not a drug yet. So the fact that no drug exists either can, of course, be a wonderful opportunity, but it may also be an indication, depending on the circumstances, that it's just not a good target. So a lot of, as you all know, negative data typically doesn't get published. Well, add in a layer of commercial research that is being done in companies. Company doesn't want to tell all their competitors that they were working on a project that didn't work. And it sounds a little bit cynical, but they'd rather have another company invest their dollars into the same project because they know then they're spending their money on something that's useless. So this actually did prompt a lot of discussion that, especially in the pre-competitive space, as they call it, so maybe there should be an impetus for at least disclosing what targets didn't work, what programs didn't work, so that there's just not a lot of waste in resources and that everybody, on a global scale just knows, okay, somebody's tried that, it didn't work, and especially if it was done in a correct way. If you get a no with the right experiments, that's really valuable information, right? So yeah, but we get a lot of times people are like, yeah, nobody's done it yet. Let's do this. And then we're like, okay, let's kind of evaluate why nobody's done this before. And what's kind of interesting too, I come from a pharmaceutical science background, more biology, but Mark is a chemist, so he looks in very different literature. I look in PubMed, as most of you probably do as well. You may Google something. He looks in the medicinal chemistry literature. So many times there's information there on certain classes of targets, maybe certain compounds that have been evaluated that are interesting from a chemistry perspective, but never really made it into the more biomedical literature. So he can really look in a different part of the literature as well to kind of help define whether there's been work done in this particular target, yes or no. So the things to think about when you've identified a target is what type of modulator do you actually need? Do you need an agonist, so something that activates it? Do you need an antagonist, something that blocks it? Do you need a partial agonist? There's a lot of pharmacology that goes into what will give you the best outcome for the particular disease. We have a couple of programs, for example, TXNP comes to mind. It's a diabetes project where we try to inhibit the expression of TXNP in diabetes to promote survival of beta cells. The nice thing is, so TXNP usually has a basal level, and glucose induces its expression. So we've been trying to find molecules that reduce TXNP expression, but only back to basal. We don't want to knock it completely out because that's probably going to cause a lot of side effects. There's been linked to cancer development, for example, if you have a knockout mouse. So you don't want to go back to zero. You want to go back to baseline. So that's just one of the things to think about. Also, where is the target physically in the body? That will influence. Example again, is it in the brain? If so, you probably need to have slightly different properties of your drug so that it penetrates the blood-brain barrier. You need to have these considerations in mind. Also, related to this is what's the route of administration. Some diseases, an injection would be fine. Others, you probably want to have something that you should take a pill once a day. That again has very different implications for pharmacokinetic properties of this molecule. How long does it hang around? Does it need to be taken up in the gut? So does it need to have oral bioavailability? Things like that.
So,
Sure.
Regarding to the where to the target, it's also, subcellular location matters.
Absolutely. So yeah, we have a program in CD38, for example, with Dr. Van Lund. So CD38, we actually think it's in two locations. It's on the membrane, but it's also intracellularly. And we've been actually struggling with this a little bit. The compounds that we currently have, do they only inhibit CD38 on the surface and not intracellularly, or does it inhibit both? And how can we differentiate between those two? And actually, what do we need it to inhibit? So it exactly speaks to your point because, does it need to cross the cell membrane, yes or no? That influences the design of the molecule. Yeah. Does it need to get in the nucleus? Some targets are in the nucleus. Very different properties than something that's a membrane just sitting on the surface of a cell directly accessible by the bloodstream. And how will you actually screen for molecules that modulate your target? What's your readout? And Bob Bostwick touched upon this a little bit last week in a talk. You design an assay to get a certain answer, whether a compound works in your assay or not. But depending on how you set up the assay, the answer can mean different things. So we talked briefly about luciferase-based screens. If you're looking, for example, for luciferase as a reporter gene under control of a certain promoter of your favorite gene, and you want to inhibit its expression, well, then you're going to look for molecules that reduce expression of activity of luciferase. Well, it can either be because the expression level was indeed affected, but maybe your compound is actually a luciferase inhibitor. We find many luciferase inhibitors actually in screening. So you need to think through how I'm screening and what does the answer mean, and how can it then filter out potential compounds that actually don't work for you the way I want it to work. And that ties into what are your secondary assays. How are you going to confirm that the molecules in this particular example inhibit luciferase? How are you going to confirm it actually inhibits expression of my gene of interest? You could maybe do qRT-PCR or something like that to validate that your initial screen was meaningful. And something too that is not trivial are the species differences. If your molecule inhibits your favorite enzyme in mouse, you screened for mouse, but then there's no homology with human, you may have a problem. Hopefully there is homology enough that you would imagine that, for example, enzymatic inhibitors would hit all species, but you don't know until you test. So knowing that information on the front end, it really gives you confidence that it's a good idea to go forward with the project. And what animal models are available? Are there standards or known compounds that you can use as control? That's very helpful, especially if there's maybe already in the clinic, something in the clinic for that particular disease. Maybe you can use that one to show that, okay, in my animal model, if I have this drug, I should see a therapeutic effect. So I'm going to test my new drug, and if I see the same effect, I have more confidence that this is meaningful. But how predictable is that to the human situation? And we talked about oncology or Alzheimer's examples already. And having biomarkers, that's really hot topic the last 5 to 10 years. More and more companies are going to look at biomarkers as a quick readout for efficacy, especially when you look at diseases where the way the disease progresses takes maybe years. So you're thinking of something like Parkinson's or Alzheimer's again. You're probably not going to get a quick answer whether your Alzheimer's drug works or not. So these trials are really long-term. They take years and years and years. So if there's a biomarker that you can identify, it gives you a quick readout. At least if your molecule engages the target or not, that will be really helpful in the clinical development. And if that biomarker also exists in the animal model, so you can do your animal model a little bit quicker, that's helpful as well. We run in this as an example now with the LRRK2 program. You may have heard about this. It's a Parkinson's disease project. There's actually a lot of companies working in LRRK2. We have the expert on LRRK2, Andy West, here on campus, so we decided to have a program as well. We started this a while ago. But now we have molecules that get into the brain. They have appropriate pharmacokinetic parameters. What animal experiment are you going to do to show that it cures Parkinson's? It's a really hard question because the field as a whole, there are a bunch of animal models out there, but how relevant are they to the disease? And the Michael J. Fox Foundation is sponsoring a lot of this research where they bring all these different companies together, commercial, but also academic, where they bet these ideas around. And one of the lessons from these summits have been that many companies don't actually put any value at this point on animal model efficacy. They rather show that it's not toxic, that it inhibits LRRK2 in the brain. I mean, you can measure that in any mouse, and then just go straight to the human experiment. Because the predictive ability of these animal models has been so poor, they rather just bypass it. So we're kind of playing with that ourselves now. Do we want to do the animal experiment, yes or no? Because we may get an answer that shows that our molecule doesn't really work that well. There's a Merck compound that we're using as a positive control. It's in the literature, in the patent literature. We have that one. Well, that particular molecule works great in monkeys, but not so good in rats. Well, are we going to do a rat experiment then and use this as a control, whereas the control is not supposed to work that well in rats, but if it works great in monkeys, people with clinical development put more weight on the monkey experiment before going to humans. But doing a monkey experiment, now you're talking a lot of dollars. So there's a lot of gray areas. There's not really a black and white, easy path to follow typically to clinical development. There's a lot of factors that go into these types of decisions. Now, luckily, we do have a biomarker, at least a little bit, or we're working on a biomarker for LRRK2 engagement because you can measure it in a urine. So you can give, an animal or ultimately a human a compound that inhibits LRRK2, and you should get a urine sample, and you should be able to tell whether it inhibited LRRK2, yes or no. Now, whether that ultimately translates into curing Parkinson's, nobody knows yet. So how many targets are there? So this is from some overviews from about a decade ago. If you're thinking of 30,000 targets in the human genome, there's somewhat of a consensus that maybe a tenth of that is druggable, and we'll talk about what that means in a little bit, so about 3,000. Now let's say also about 3,000 are related to some sort of disease state. You're looking at between 600 and 1,500 targets that may be a good idea to go after. And currently, targets with FDA-approved drugs, there are 324 targets, if the target is known. We actually have a lot of legacy FDA-approved drugs that we actually don't know how they work. But for the ones that we do, about 1,500 drugs that target a little bit more than 300 targets. So there's still new targets to explore and develop and analyze. So what are those targets then? Examples. So many times people look at receptors and enzymes, so protein-based targets, either, as Shu mentioned, intracellularly or maybe on the membrane surface. GPCRs have been targeted a lot, G protein-coupled receptors. Kinases, proteases, phosphatases, so these are enzymes. Transcription factors, nuclear receptors, ion channels, and transporters. Ion channels are very heavily targeted as well. And then what used to be called, more difficult, more undruggable protein-protein interactions, because it was thought if these proteins bind to each other with a lot of hydrophobic surfaces, it's really hard with a small molecule to disrupt that. But there's actually more and more examples now that we can do that. We actually, the tau-fin program in the ADDA is actually a protein-protein interaction program. And then you can do what we call phenotypic screens, and an example of that is the luciferase-based screening system I just talked about, where you just look at a particular readout of a cell. Is a cell gonna glow or fluoresce or die, or does it grows out dendrites, things like that. So looking at some change in the cell as a whole after you administer a drug to it. It's called a phenotypic screen because you add your drug and you look for a favorable outcome. The problem is in those types of screens, how do you know how the molecule works? You don't really know what the target is. Sometimes you're looking for maybe, like we said, an increase in the gene level expression or activity of a certain enzyme. But if that's the outcome, you really don't know what the drug itself is doing because you add it to the cell and against a black box, it can work on many different levels to ultimately lead into outcome. And even though the FDA doesn't require you to know the target, it's still a really good idea to do know it because it will influence how you're going to design your clinical studies, your preclinical studies, and so on. Because if you know exactly what type of molecule it targets, then you're going to get a sense of what potential toxicities do I need to be looking for. If it's something that's maybe widely expressed throughout the body, maybe it's not that good of an idea to do this program. So knowing the target is at some point going to be a critical question just from a commercial, or maybe not even commercial, but just from an intellectual standpoint of, is this a good idea to pursue, yes or no? So here's a graph of what current drugs target. So about a quarter are the GPCRs, like I talked about. This yellow one, 13%, those are nuclear receptors. Then these are ion channels, also different types of ion channels, and then the pieces of the pie get progressively smaller. So there's a lot of experience with GPCRs, so those would be considered highly druggable. Another example are things like kinases, because they're being targeted a lot, they're being used in oncology a lot, so those would be called a druggable target. So how do you then identify what targets, may be a good idea in a particular disease? So historically, people would have used natural products from, less civilized other civilizations and things like that, where herbal medicine or maybe something like snake venom was always treated for a particular disease. If you know what those natural products actually interacted with, then maybe you can design a drug like it to have the same effect. Now, sometimes clinical observations lead that if you have side effects of known drugs, those side effects may actually sometimes be a useful outcome in certain disease pathologies. So you can then repurpose those drugs for other indications. But most of the times, what we run into is rational approaches based on the biochemistry or the biology of a certain disease. And then especially if you have genetic mutations in people that play in a particular pathway, that have a particular propensity to heart attacks or, hemophilia or you name it. If there's genetic mutations in people that have the same outcome as if your biology in the cell culture system has shown, that gives you a good indication this is a relevant target. And then nowadays, a lot of people are doing screening in systems biology to find new targets, just screen lots of databases and see, okay, in tumor tissue, are the things highly expressed compared to normal tissue? And if so, maybe those are good targets to go after. That we kind of talked about on this slide. So the omics technology, so genomics, proteomics, pharmacogenomics, now probably the microbiomics, all those types of technologies, RNA screening and pathway analysis, transgenic analysis, they've all resulted in a lot of targets to potentially go after. It also gives you a little bit of information overload, though, because there's so many potential targets that get spit out by these types of screening technologies. How do you know which one to go after? Again, so the key question always remains, how are you going to validate that this is a good idea? So here are just some validated target examples with the question mark, of course. So with respect to genetic mutations that are associated with disease, here comes our Alzheimer's example again. So there has been some genetic hints at that changes in amyloid precursor protein, or APP, in secretase that cleaves this APP and results in A beta, that's leading to Alzheimer's. Well, there's genetic information, we have animal models that do that, but we still don't have a drug that actually is able to target it and reverse Alzheimer's. So whether it's validated or not, that's an open question. HER2 in breast cancer, there's naturally occurring mutations in HER2. Again, that then resulted in development of antibodies in this case that target HER2, and it actually is resulting in better outcome in breast cancer patients. So that was what we call a validated target. Kinases are pretty validated. There's a lot of kinase inhibitors these days. And I just want to give you another example. Sometimes certain pathways or enzymes are upregulated or maybe downregulated in disease processes. So an example is inflammation in COX-2, cyclooxygenase 2. So classically, they had, NSAIDs, non-steroidal anti-inflammatory drugs, are COX inhibitors, cyclooxygenase inhibitors. They were not selective, so they inhibited all the COXes, which resulted in a lot of side effects. So you can get irritation of your GI mucosa, which sometimes is based on the mechanism. I'll talk about it in a second. And also sometimes the properties of these drugs themselves are very acid-like, so they tend to be very irritating to your stomach. And that's why you probably don't want to pop, ibuprofen constantly, because you're going to get GI issues. So in the 90s, they discovered that there's actually different isoforms of cyclooxygenase. There's 1 and 2. So COX-1 is present in most of the tissues, and including in the GI tract, where it plays a protective role. And that's why when we take something like ibuprofen, it inhibits both COX-1 and COX-2, so then this protective lining in your GI tract gets damaged, and that's why you get the side effects. Now COX-2, that's kind of the bad actor. That's the enzyme that gets upregulated in inflammation. So then the concept in the 90s was, let's find molecules that inhibit COX-2, but not, that's a typo, but not COX-1. And that makes intuitive sense, right? Get the benefit without having all the side effects. So everybody jumped on this bandwagon, and they developed COX-2 inhibitors. So celecoxib or Celebrex from Pfizer is an example. Rofecoxib or Vioxx from Merck was another one, and they kind of raced each other. So Vioxx got on the market, and then it was actually voluntarily withdrawn in 2004, so because there was an increased chance of heart attacks. So before the FDA made Merck pull it from the market, Merck actually volunteered, like, okay, we see this data too, let's pull it from the market. Well, actually, there was an advisory panel that was put together the year after that, and they looked at all the data and also the Celebrex data from ibuprofen, et cetera, and they said, actually, when you compare the risk factors for cardiovascular diseases in something like ibuprofen with Celebrex, it's the same. So it was just always underappreciated that things like ibuprofen would actually raise your risk of heart attacks. So Merck actually did not put it back on the market, probably for PR reasons. I don't know. There was a lot of backlash to the pharmaceutical industry when this all happened. I don't know if you remember it or not, but you got the usual pharmaceutical companies are out there just to make a buck, and they're screwing over the population, and they suppress data, which turned out they actually had not. It was a honest mistake, sounds kind of wrong, but there was not a cover-up of this data. It's just it was not appreciated until Vioxx was prescribed to these thousands and thousands of patients that this happened. Now, actually, interestingly, December of last year, so only 4 months ago, This company, Tremo Pharmaceuticals, actually is thinking of returning this back to market now. They have, they talk with the FDA and starting a phase 3 study for joint pain and hemophilia. So a very targeted indication because it does work to treat the pain and it doesn't have as many side effects, especially in the kidney actually, as something like ibuprofen. And this is just a magazine. When this all happened, so 2005 with the advisory panel, they actually got together with all the industries and the FDA and so on, and they said, Pfizer, the Celebrex guys, if you want to, be good, fund this $500 million study where we're going to compare Celebrex, I think naproxen and ibuprofen. So this study, I think it was called Precision. The data came out, about 2 years ago now, a year and a half. So actually Celebrex was as safe as ibuprofen and naproxen, and it did have significantly less GI side effects and also less kidney side effects. So, but it took a study done over, I think it was 25,000 patients, again, $500 million to actually to demonstrate this particular safety profile. But in the meantime, the damage was done to the Vioxx drug. So then you get into, okay, so is COX-2 a validated target, yes or no?
Yes.
I would argue so. Now for Merck, was it a viable target? Would have been a good idea for Merck to come back on the market with a COX-2 inhibitor. Maybe for business reasons, not so much. So again, there's different aspects to different targets. So when would you then consider a target validated? Is it when you first do your mechanistic studies that shows, okay, I inhibit this enzyme and I get this particular outcome? Is it when you analyze in cell culture? Is it when you do your animal models? Is it when you have clinical data? Is it when you show proof of concept in phase 2 studies, or is it not until you have FDA approval? I don't think anybody knows. Probably everybody would agree this is too early. This probably still is too early. If you show efficacy in animal models, you get maybe a little bit more confidence. Somewhere in this bottom half of this slide is probably where the sweet spot is, but it's really hard to give you hard and fast rules when you consider target validated or not. So some operate under the assumption until you show that the drug works in humans, it's not validated. But if you only wait until that point before you start a drug discovery program, you're always going to be behind somebody else's, right?
I want to comment this. Sometimes if a compound has a problem, and that's cannot exclude that this target is not good. Because if the side effects are compound associated, right?
That's exactly right. So when you have a bad outcome, for example, in your animal model, is it because your biology is wrong, or is it because your compound's not good? Sometimes you don't know yet, and I actually have a slide on that exact issue, which actually is coming up now. So if you don't follow in the pipeline yet, this is one of the blogs I read. It's wonderful for drug discovery. Derek Lowe, he used to be, he was working for Vertex. I think he got fired or quit last year. I'm not sure. But it's now on the Science Translational Medicine website. He publishes, 2-3 times a week or so about drug discovery in the pharmaceutical industry. So it's very low threshold, easy to read, quick stories. And this one, I remembered, was about target invalidation or validation. It's about 2-3 years old. And he was referring to a paper that came out in American Chemical Society Medicinal Chemistry Letters about target invalidation, critical, sometimes unheralded, role of modern medicinal chemistry. And unheralded is probably usually because here's the chemist and the biologist, you were wrong, which typically doesn't go over that well, as you may imagine. But I just highlighted some of Derek Lowe's musings on that. And this is the top part is from the paper. And it says, too often, medicinal chemistry teams blindly accept the biology of a new program as sound, and that they are unaware that their efforts at compound optimization may either confirm or debunk the link between the target and the disease. And in many instances, this event occurs very early in the drug discovery process, usually preclinically. So that would actually be good if you can, at that early on, say this is not a good idea. It saves people, time, money, and so on, right? Because then you can go to the next problem. But Derek comments that actually the number of targets that you can be sure are good ones before anybody actually has made a drug is not large. So then he talks about an example called PCSK9. It's actually, there was a genetic mutation found in humans that people who had this mutation had a higher risk of cardiovascular disease and heart attacks. So then a lot of companies went on to find inhibitors of this enzyme to lower LDL levels, kind of a new type of statin. So they actually found molecules, different companies found molecules that inhibit PCSK9, and indeed in clinical trials it lowers LDL levels, so it's a good outcome. Now the problem is actually more recently, when they looked at, well, do I actually then prevent heart attacks? The answer is no. So again, is it validated or not? It lowers LDL. Everybody until a few years ago would probably be like, yeah, that sounds good. And then when you look at outcomes, maybe not. Anyway, the jury's out on that. But here he says a little bit later, there are a few targets kicking around out there that have never really been drugged, but everybody's pretty sure will do a good thing, like P53 or MYC in cancer. Those are elusive targets. A lot of companies have been trying to inhibit P53 and MYC, and everybody agrees if you can inhibit those, you're golden. But otherwise, unless somebody else has already blazed that trail, you're going to be finding out not only is this compound going to hit the target, but also is this target worth hitting in the first place? You typically don't know until you do the experiment, right? And then here from the paper again, it kind of stresses the infrastructure behind every program should be stress tested at a very early stage. And this is, I think, the key. Fear of obtaining suboptimal results from a suboptimal compound should not be a reason to defer such testing. So that speaks to your comment, Shu. If you don't have a good compound yet, do you really want to test it or not? Let's see. The feedback to biologists is quite helpful to establish whether the assay you're looking for is a good idea or not. And so here Derek kind of comments, oh, if you try to test using just the chemical matter we have now, you'll probably kill the project for no good reason. So again, okay, let's not do it yet because our compound's not perfect. So it's not actually a silly argument. It is possible to go in too early with something that's not potent enough, for example. But you can also use it as an excuse to delay, he calls it the day of reckoning, to avoid getting an answer that no one wants to get. You have to be honest and be ready to hear bad news and not keep telling yourself that you're waiting for a better compound before asking the hard questions. Otherwise, you have a compound that's been optimized and is ready to go to the clinic before you even know if you should have been doing all that work in the first place. And then just the last little excerpt. Let's see. Even trying out your compound in vivo and seeing the effect you want may not be enough. It's a big step, so it's a good idea, but you'll be a lot more confident if you can have this effect with more than one chemical series. So again, that will speak to what you just brought up. Maybe they're just a side effect or of a certain compound series. So if you have a different one, it's the same target, but very different structural scaffold, then you have a lot more confidence that your target is a good one and that, you may not have side effects that, are target related. Let's see, and he finally suggests take some structurally similar compounds. So if you don't have the luxury of having two series, then use the series you have your best compound and go in it. But usually you'll also have similar compounds in the same series that don't work at all. Use those as a negative control. And it's those negative controls, we always tend to gloss over them when we design experiments, but that's absolutely critical. And he also does say, it's a good advice, but it can be a tough sell with some biologists. From what I've seen and from a look over the history of drug research in general, I'd say that skimping on the negative controls is both one of the most common shortcuts and one of the ones you'll be most likely to wish you hadn't taken. So I think that's really sound advice, and especially the negative control idea, I think, is something that we'll incorporate in the ADDA. So here then some of the criteria when we look from an ADDA perspective, whether we want to take on a target or not. So is it druggable? As in, is there evidence that supports the drug discovery program? Are there small molecule inhibitors for this class of targets? There may not be one for that particular target, but maybe a sister or brother target has been targeted with a small molecule. What's the availability and predictive value of the animal models? And are there species differences, and is there a biomarker available? Do we have a crystal structure? Do we know NMR structure? And what's the IP space? What's the competition? If everybody's working on a certain target, especially pharmaceutical companies, then maybe we as an academic institution don't want to play in that same space. Unless there is a really compelling reason for it because we have a different approach or we have the world's expert on this target on campus or something like that. And it's not like this, you have to answer yes or no to every question, but it kind of comes into, when we compare different targets, we look at the answers to these questions to evaluate which one we want to invest in, yes or no. Because there's never going to be probably a perfect target that you have all the information here, so you make some educated guesses, but you can probably compare 2 or 3 and decide, okay, we should put our eggs in this basket because it's probably higher likelihood of success than the other ones. And just some examples. So we used to do phenotypic screens, where again, you have this setup of a promoter of a gene of interest that drives luciferase. HO1 is a program with Dr. Agarwal, 1433 theta is a Parkinson's project with Jacobian, TXNIP with Dr. Shalev, and an NMI with Dr. Samant. But the problem in all of those screens, we ran screens, we found compounds, and especially in the TXNIP program, actually, we have molecules that we've done toxicity profiles on. We've been able to show really good efficacy data in animal models of both type 1 and type 2 diabetes. We don't know how it works. And it's a real problem going forward, again, because it will inform maybe toxicities we need to look out for, maybe counter screens we need to do, designing of the clinical experiment, and so on. And also, I've talked about it in separate lectures, at some point you want to collaborate with somebody who has lots of money, right? Pharmaceutical companies, we've talked to some companies about some of these programs, and they say, sounds really interesting, your animal data looks cool, call me when you know what the target is. Before that, they don't want to play with you. So, shoot.
I have questions. For example, if you get a compound from these phenotypic screens, so you know works, so they is it easy to identify?
No, it's really hard. So the approach that most of the programs are taking now is doing something like RNA-seq and see what pathways are going up or down, and based on that and the knowledge of biology that we have, identify what the target is. And sometimes we do have a little bit of a sense of, okay, it's probably, for example, with the TXNIP program, we know it has to do with glucose control. We know what pathways are involved in that. We kind of have a little bit of a sense, but to pinpoint it exactly, you're going to need to do a lot of experiments with, knockout models and IP, co-IP, Western blotting, things like that, to really hone in on it. So it's very difficult, and that's why we, as a program, didn't really realize this when we started. Now, in retrospect, you're of course like, well, duh. But when you go forward, you restart something you don't know. So actually, right now, we won't take any programs anymore in the ADDA that are phenotypic screen because of exactly this problem. Because again, we have molecules in pretty much all these programs that work fantastic, but we cannot find a partner until we know what the target is. And we can only do so many programs internally with our own dollars, and then there's the question, do we really want to invest in a program where we don't know what a target is either? Because we may run into issues later with the FDA. Now, we do have a lot of defined target examples. CD38, what I talked about earlier with Dr. Lund, it's on the surface, but it's also a pool intracellularly. So it's a really direct target. DPY30 is an example. That actually is a different way of getting at MYC. And we ran a screen where DPY30 binds to certain initiation factors that have to do with chromatin modeling and so on. LARC2 and taufin. So in those kind of examples, we really know exactly the protein that we want the molecule to bind to. We know what we want the molecule to do, and sometimes we even know where on the molecule we want, where on the protein we want it to bind. And that's just it's very helpful information, especially if you have maybe a crystal structure or NMR. DPY30 is a target that Dr. Will Plazik is involved with, and he does a lot of NMR studies. So we're actually supplementing a lot of the high throughput screening with just shots on goal and sifting through the haystack to find a needle with an NMR-based screening system, so we can identify what amino acids is this molecule actually binding to. Oh, that's it. So there you go. Next week we have Ed Acosta coming to talk to us about pharmacokinetics. But for now, I'll stop and we'll be happy to entertain any questions. And if you have any cool targets, just contact me. Questions, concerns? No? All right, then I think everybody needs to duck out early and have a long weekend.
Oh, I have a question.
Yeah, shoot.
You said it's difficult to find identify target.
Yeah.
So is it possible I do 2D gels, or radio lab or the compound, then put in the cells, you know, or you know, do 2D gels, identify spots with high radioactivity. And you just pick up the spots in next step.
Yeah, I think that may be one approach. It sometimes gets a little tricky how you radio label molecules, and also sometimes people have suggested to do click chemistry. To click chemistry is a certain type of chemistry where you add a tag of some sort to the compound, and then you can do screening. Different companies offer this to see where and then you shine at the sample with UV light, I think, and it changes and it forms a covalent bond to whatever it's close to. Or you can use some gel chromatography type things to see if you have the compound on the gel material in the column, and then you run a lysate to it, what sticks to the compound. The problem is when you manipulate the compound to have that linker, you change the structure. And it depends on how well you know the structure-activity relationship, whether that is tolerable or not. Will it still have the same effect, yes or no? So you need to do actually a lot of work to even get to that point. That isn't an approach that some people do. It's just to get there is more tedious than sometimes people realize. But yet, radioactive labeling and other types of labeling is one of the approaches. You looked at outside vendors to see if there was a company or companies that would offer some of this. You know, we'd be happy to pay somebody a lot of money, figure it out, but most of them also use RNA-seq and just use software to analyze the data, and it spits out options. It doesn't confirm for you what the real target is. It just say, there's a likelihood that this is the target, or maybe this one or this one, and we kind of rank them, but they still don't really know. So it's complicated. Yeah, if you have ideas, I think many people would like to hear them. All right. Well, thank you guys. Have a nice Easter weekend, and hope to see you next week. And again, if you haven't flipped your card, please do so on the way out, because we need to check our, reimbursement and all that kind of stuff. All right, let me stop the recording. Oops.